Hi everyone, I'm Mina Karimpour from Cancer Research UK National Biomarker Center, and I'll be presenting our recent work on multi-omics analysis of cell-free DNA for EpiNEC diagnosis and treatment monitoring. Extra-pulmonary neuroendocrine carcinomas are rare but aggressive tumors that originate outside the lung tissue. We know that the current treatments fail to address the significant biological and genomic heterogeneities in EpiNECs, which highlights the urgent need for biomarkers to optimize patient management. In this regard, cell-free DNA, which is a free-floating DNA in the bloodstream, has emerged as a promising source of cancer-related genomic and non-genomic information. This allows us to have a multi-omics approach for biomarker discovery. So, in this project, we aim to explore the potential of cell-free DNA multi-omics profiling combined with a computational workflow for EpiNEC diagnosis and clinical management. For this, in the first step, we collected blood samples from 53 EpiNEC patients at different time points and 79 non-cancer controls. After plasma extraction, cell-free DNA profiling was performed using T7-MBD-seq and methyl capture sequencing approach. In this method, 10% of sample is used for shallow whole genome sequencing and 90% for methylation enrichment profiling. With these two levels of information, we defined EpiNEC cell-free DNA biomarker modalities. We began with copy number analysis using the well-established ISMB tool, which estimates the circulating tumor DNA fraction using copy number information. We analyzed cell-free DNA fragment sizes, focusing on the ratio of the short to long fragments. This is because cancer samples typically have a higher proportion of shorter fragments than normal samples. For methylation profiling, after pre-processing of the data and estimation of scaled methylation beta values for 300 base pair windows, we identified differentially methylated regions between a subset of pretreatment EpiNEC and non-cancer control samples, and then validated in the remaining samples. Then for each sample, we calculated methylation score by averaging beta values over the identified DMR windows. Given the histopathological similarity of EpiNECs to small cell lung cancer, we decided to use our in-house small cell lung cancer healthy classifier to detect the tumor signal in EpiNEC samples. This classifier has been trained based on cell line-derived or patient-derived xenograft models using ensemble of XGBoost classifier. The tumor prediction score threshold of 0.25 was used to detect tumor signal in EpiNEC patients. By this computational workflow, we were able to define 4 EpiNEC cell-free DNA biomarker modalities. From genomic features, EpiNEC samples exhibit more copy number alterations compared to non-cancer control samples. Translating this copy number information into icosCNA tumor fraction shows that some pretreatment samples fall below the 3% threshold and unable to be detected by icosCNA tumor fraction. From fragmentation feature, as we expected, the short to long fragment ratio is significantly higher in EpiNEC sample, and this ratio is correlated positively with icosCNA tumor fraction. As I mentioned, we have validated the identified DMRs in the validation samples. This heat map of methylation beta values shows that EpiNEC samples almost clusters together with hypermethylation in the DMR regions. We can also see that EpiNEC methylation scores, which are the average of beta values across these DMR regions, are significantly higher in EpiNEC samples than non-cancer controls across all 3 time points. There is also a positive correlation between methylation score and icosCNA tumor fraction. Here we did a longitudinal monitoring for 6 patients with additional follow-up samples and observed that EpiNEC methylation score can detect the dynamic variations in tumor burden that aligns with the changes in disease volume from CT scan images. So this cell-free DNA-based biomarker could be served as a quantitative measure for clinical associations and EpiNEC monitoring. Application of a small cell lung cancer healthy classifier to EpiNEC samples shows that almost all the baseline samples have a tumor prediction score above 0.25 cutoff. This signal decreases 6 weeks after treatment and rises again at disease progression. Comparing the iCO-CNA tumor fraction with tumor prediction score, some samples that were undetectable by iCO-CNA were successfully classified using our methylation-based classifier. This indicates that the tumor prediction score outperforms the iCO-CNA tumor fraction in sensitivity and specificity. So in summary, we were able to capture multiple cancer-specific cell-free DNA features using our T7-MBD-seq method. We saw that methylation profiling increases the sensitivity of EpiNEC detection compared to using genomic profiling alone. We also demonstrates the potential of multi-omics cell-free DNA profiling and machine learning in EpiNEC patients for biomarker discovery. Our future work will focus on increasing the sample size and developing the EpiNEC specific classifier to improve diagnostic accuracy. And now I finished my talk, and I would like to thank the entire team for their contributions, as well as the patients and their families.